The inventor, Dr. Tatiana Coelho Sampaio, UFRJ, Brazil. A biologist at the Federal University of Rio de Janeiro, UFRJ, Tatiana Sampaio has spent 25-plus years studying a humble but extraordinary protein called laminin. Laminin is naturally present in every human body. It acts like a molecular scaffold guiding nerve growth during embryonic development. After birth, it becomes much rarer. Her key insight: what if we could recreate that scaffold in a lab and inject it directly into a damaged spinal cord? We are simply imitating nature, since this protein is naturally produced by the body during the development of the nervous system. Dr. Tatiana Coelho Sampaio, UFRJ. Research started, 1999. Institution, UFRJ, Brazil. Royalties to UFRJ, $3 million R+. Patent acquired by Cristália, 2022.
The science: a molecular scaffold that rewires broken nerve paths. Spinal cord injury: axon severed, signal path broken. Polylaminin injected: single intraspinal dose within 72 hours of injury. Nerve regrowth: axons grow new paths, signals restore. How the axon bridge works: polylaminin scaffold, new axon pathway, re-established signal. Polylaminin is a polymerized form of laminin. Basically, laminin molecules linked together to form a stable 3D mesh structure. This mesh acts as a molecular scaffold at the injury site, stimulating neurons to regrow their axons, the long wires that carry signals between brain and body, through the damage zone. The key detail: ordinary laminin doesn't work therapeutically. Only the polymerized version creates the structure needed to guide axon growth. That's the core invention: figuring out how to polymerize it correctly from placenta-derived protein. Source material: human placentas. Administration: single injection. Treatment window: within 72 hours. Dose: 1 microgram per kilogram.
This is one of the most fascinating stories in modern medicine, and it's happening right now in Brazil. The research was led by Dr. Tatiana Sampaio, a biologist and professor at UFRJ. She discovered that laminin, a protein the human body naturally uses to guide neuron growth during embryonic development, could be recreated in a lab by extracting proteins from human placentas. She figured out how to polymerize it, link the molecules together, into a stable 3D mesh, and that polymerized version is polylaminin. Digital origin tech. What happens in a spinal cord injury is that the communication between two neurons, one in the brain and one in the spinal cord, is interrupted. What polylaminin does is help the longest part of the neuron open a new path at the site of the injury, generating the electrical impulse necessary to perform a movement. The human pilot results were extraordinary. 6 participants regained some degree of voluntary motor control after receiving a single injection of placenta-derived polylaminin directly into their spinal cords, within hours or days of their accident. Bruno Drummond, the most dramatic case, went from complete quadriplegia to walking within months. Phase 1 clinical trials were authorized by ANVISA in January 2026, making this very much a story still unfolding. If it clears all 3 trial phases, it would be the first drug in history to restore motor function in complete spinal cord injuries, a category with no approved competitor anywhere in the world.